All right, so welcome everyone to this session. My name is Ilya Brodsky. I'm CEO of Pine Biotech, one of the co-founders. And I want to briefly tell you about bioinformatics in general. I'm sure that you all have different backgrounds. So I'm really ready to learn more about your background and your interests as well. And hopefully we will be able to do that throughout the meeting together. I would love to, know where you're from, which country, what is your interest, what degree maybe you have at your university, and where are you headed? Why did you decide to join this particular webinar or this program in general? So as we go through the next few minutes, we'll start looking at how we can make sense of it all and kind of find a right place for bioinformatics and data science and understand what opportunities lay ahead for these disciplines if we are interested in biology, biotechnology, etc. So what is my background? My background, when I was, a young adult, I was in an applied mathematics. The reason I went into applied mathematics originally was because of my parents. My father is a mathematician and a statistician, and he thought that this was the right way to go. And so I decided to join that program. Unfortunately, very quickly, I realized that that is not exactly what I want to do with my life. And I quit that program. Fast forward, 10, 15 years later, I realized that actually everywhere you go, whatever field you are in, data and understanding of how to manage and analyze this data is critical. And so today I have over 8 years of experience already with bioinformatics and have been a part of many projects that are very interesting, but would not be successful if they did not have a data analysis and a data management component. So I'll briefly talk about those later throughout this session. But before we jump into that, I want to kind of give you a little overview of what I hope to cover in today's session. So first, I'll talk about the role of data in the research lifecycle, how it is important to know what kind of data is going to be generated, and it is important to know how to then analyze and interpret this data in the context of an experiment. I also want to talk about the role of the person who does that, and in the context of, whether you are exclusively focused on just data analysis or you are actually focused on combining different skills of biology and research and bioinformatics or data science. And then I'll talk about how to get started. So what I find with a lot of people that get into bioinformatics for the first time, they are being told in a very traditional way to get started with very simple things. And on one hand, it is the right way to do it because you need to learn the basics before you move into more advanced things. But on the other hand, it is also intimidating for people who have a lot of experience, who are, very smart, who are passionate about what they do, because it's irrelevant, right? And so there's a fear of losing time, of doing something irrelevant, of not really understanding how what I am learning fits in with what I really want to do. Then I want to talk to you about your future career. You might be thinking of your academic career, or you might want to go into industry. And so we'll talk a little bit about what are people looking for, right? What are those skills? What are those types of experiences that potential employers might be interested in? And how can you leverage what you do with OmicsLogic in those discussions, in those plans? And that brings us to kind of the availability of many different resources online that you could potentially go into that have, excellent lectures, different kinds of coding exercises and tutorials. So really talk a little bit about, how to organize those kinds of things in a way that it can actually help you build up your background, your portfolio, and what does that mean? How does that practically work? And then finally, we'll talk a little bit about our community, because this is more than just a discussion about training or acquiring skills or even developing a research project. Really, we are here today because there is a global community of people who come from different backgrounds that are interested in life sciences, but also data. And to build up this community, to participate in this community, you need to be able to give and take at the same time. So hopefully we'll talk a little bit about how you can contribute and how you can benefit as well. So let me start with kind of talking a little bit about molecular biology in the context of precision medicine. Recent innovation with biotechnology and biomedical research has led to these high throughput technologies, right? And these high throughput technologies allow us to understand what is happening at the subcellular level, but it is reliant on a parallel development of computational technologies. And those computational technologies are potentially useful if they could be applied to solve these data analysis gaps. And really, this gap between data and information, even though more and more people acquire data science skills, continues to grow. Now, that leads us to the question, why? Why, even though we have all of these technologies, right, in the lab, we have these technologies in the computational world, why does this gap continue to grow? And the reason why it continues to grow is because these technologies in the hands of experts allow them to generate data quickly in an automated way, right? What does it mean in an automated way? That means that you go home and the machine continues to generate new data, right? So when you look at it that way, it really means that, there's a handful of experts that are able to now analyze this data, and the rest of the people are waiting for those experts. And that creates a bottleneck. And that bottleneck leads to an ever-growing gap between the amount of data that is available and the information that we extract from this data. In a recent survey, they asked researchers what kinds of steps they are more or less familiar with in the context of next generation sequencing. And it turns out that the majority of the people, more than 50% today, that was a while back now, but today more than 50% are intimidated by the data analysis part, because really, data analysis is a process. It's a process that includes exploratory analysis. It includes kind of thinking of the specific contrasts and comparisons that we can make. So a lot of people are actually intimidated by this process of not being able to analyze data effectively. And so what we see as the real challenge here is the challenge of bioinformatics in general. It's an incredibly dynamic field with a lot of changes. You probably all know, Illumina was at some point was not even around, right? And so everybody relied on Sanger sequencing and microarrays. Then Illumina came along and everybody jumped on Illumina. And today, Oxford Nanopore is kind of the emerging technology with a little bit of a different technique to generate data, right? So this technology continues to evolve in a fairly rapid way. There's always new packages, new scripts, new ways to visualize data. And so you have to be understanding of how all of that ties together. The universities where a lot of this research happens, they have a limited amount of computational resources that are available to everyone to use and to store that data. And also students, even though they understand theoretically what needs to be done, it's a time-consuming and very complicated process. So you really need experience. And then finally, I think that, we look at faculty who are supposed to be teaching students how to do this, but they really have limited experience themselves, and there's just not enough talent for teaching effectively these skills. And so to address these challenges, what our team has done is we have started building out the OmicsLogic portal. And the OmicsLogic portal is designed to standardize, organize, and systematize this information available on bioinformatics training. So this challenge is recognized by many around the world. We're not the only ones who recognize the challenge, but we believe that this portal that we have put together with the different resources, and I'll talk about what those resources are, really helps address some of that challenge. So we talked about the role of data. We talked about, how to kind of see this, right? And now let's talk a little bit about the psychology of getting started, overcoming the fear of solving irrelevant problems. So to do this, we'll do a little bit of a practical thing. So first of all, I believe all of you should already have a profile on the OmicsLogic portal, right? The OmicsLogic portal, let me just quickly kind of walk you through. I know that many of you are probably already familiar with it, but I'll just walk you through a couple of things right here on the homepage. So this portal is designed to introduce several important kind of components that I think are very useful if you're interested in getting into this field of bioinformatics. Number one, we have mentor-guided programs. So these are experts who have a lot of practical experience in bioinformatics already, and they conduct training programs throughout the year. We have practical assignments where you can understand the logic of the analysis, maybe not being well-versed in the technical aspects of bioinformatics. There's coding exercises, and those coding exercises are kind of available on demand, which means that if you need it, you can learn how to do it. But if you don't need it, you don't need to worry about it. And then what's important is also it allows us to kind of understand for you yourself, right, to quantify your progress. So what I wanted you to do right now is to go into your profile. So by the way, let me ask you, how many of you have already created a profile on OmicsLogic? If you have, please share the link to your profile in the chat box. So the way you can do it is you can go to your profile by clicking on your email address here on top, okay, and copy this link above and share it. So this is my profile, for example. Okay, so let me know if you have completed your profile. I'll give it a couple of minutes so you all can share. And hopefully we will learn a little bit about each other through this process as well. All right, thank you. So this profile, and if you are able to, please share the link to your profile as well. This will help us connect with you, and will also help us all kind of learn about each other. This profile is what people see, right, when they find you, right? So for example, if I'm an employer, right, and I'm looking for bioinformaticians because there's a lack of qualified bioinformaticians, and someone comes to me, I would like to know what has this person done or learned, right? If I'm a potential collaborator, right? I'm a research, I have a research lab. I need to find someone to collaborate with. It's a way to show what you know how to do, right? So what's on this page? First of all, it's always nice to see who you are, right? So I uploaded my picture here, and this is actually what I looked like a few years ago. There's social media. The social media is an important part of our day-to-day life, and I recommend for everyone to have a social media profile. In the research world, if you are publishing, it's also useful to have a way to share what your publications are, and ORCID is a good place where you can list out those publications that you have been a part of. But an important community, I think, professionally, is LinkedIn. So I have my LinkedIn profile here as well. And by the way, I recommend all of you, I recommend that we connect. So if you do have your LinkedIn profile, you're welcome to connect with me. And I'll also show you our community here on LinkedIn, where we have a community of over 2,000 members from all over the world, where they specialize in machine learning, in biomedical research, and it's a really great place to find out about job opportunities, about research projects that people are working on, etc. So please join this community on LinkedIn as well. So what else do we have here? An important part here is a little blurb, a bio about you, okay? And by the way, we have our team here with us on this Zoom meeting, and they are taking notes of all of your profiles, and hopefully we will be able to review them. So the first thing that you want to do is you want to present yourself in a good way. And the way to do that is you click on edit your profile. So when you edit your profile, you can add your name or change it. A good way to change your name is to make sure that you have capital letters in the beginning and not all capital letters, right? This is important because when you get to certification, that shows professionalism, the first thing that people might be looking at. You can add here the links to your social media, and please use the full link with HTTPS and the full link of this present as well. And after you are done, your profile is going to say that it is 100% complete, okay? So if you have not done this yet, please do that today so that we can continue and stay in touch after this session, because I really believe in a growing community that can learn from each other, and this is an important part of what we're trying to do here at OmicsLogic. The second thing is you will find here under the course tab all of the courses and projects that you have taken or are in the process of completing. So you can see that for some projects, I have done just a little bit. Some of them I maybe have done more. And as you want to turn to a course that you have completed, you will also see that there's a certificate. And this certificate is something that you can share, for example, on social media, right? So here I've completed Introduction to Bioinformatics, and I can see a certificate and share it with the community, telling everyone that now I am learning how to do that. Okay, so let's go back. Now, I hope that all of you took a little bit of time to complete your profile. And by the way, if you want to see if what activity is there, you can go to the Achievements tab right here, and you will be able to see that some people are actually completing their profile right now, right? So after you complete your profile, you are given 500 points. And so you're moving ahead, you're moving along, you're maybe overtaking some people, you might be becoming more proficient than others, and it's something that we can measure. Okay, so here you can see excellent other people that have joined, and I love the fact that somebody completed their profile, so I'm going to like it right here. Let's go back to our presentation. So how to get started, overcoming the fear of solving irrelevant problems. Let's talk a little bit about the process of learning. So the process of learning is not something that just happens by itself. In fact, there's a lot of research going on into the process of learning and how you become proficient at something. And I hope that all of you have thought about this as well. And I definitely have, because when you are starting with something new, and bioinformatics might be a new topic for many of us here, that topic is, when you speak to an expert in that topic, it's almost like speaking a different language, right? It's almost like you are, speaking Chinese all of a sudden, because every single word you need to understand, what does it mean? So the process of learning starts with acquiring that language and that, lexicon of that domain, kind of understanding the basic concepts, understanding the facts, knowing what ideas or concepts are being used. And as you build up, right, so if we think about it as a pyramid, if you kind of start building up on that foundation, eventually you are able to use that information in a practical way. You can apply it. And when you read a publication, you can start drawing connections between different ideas. And then you can start thinking critically about someone saying, I did this like this, or I did it like that, right? So you are starting to really grow in your understanding. And eventually, at some point, right here, you can start creating new or original work. So thinking about this process of going from kind of understanding the basics, building on that, to apply that information in a guided way, and eventually becoming more and more independent and practical, we have started thinking about that process and thinking about how can we make OmicsLogic built around this process. And so this is what we did, right? We created a portal that brings together resources that are of different levels. They have practical things, they have conceptual things, and they also have mentor-guided, more kind of open-ended and creative aspects as well. And that's really the message of today's meeting. Hopefully this is something that you can take away from today's session, is a better understanding of how this resource can be helpful to you and what else you can do with it besides just joining a few sessions and looking at a few online courses, which is, of course, what you can find on many other places as well. So let's talk about how to, do this in a way that you can actually gain something out of it. So we said the first place is to say, I'm a part of this community, create your profile and complete it, right? Upload your picture, give us a bio. What are you interested in? What are you doing? What is your background? What is your educational level? And then connect us to your life, to social media, right? To your professional profile, to your academic profile, etc. And then you want to start somewhere, right? So where do you start? If you go to the courses tab right here, you will find over here several different ways to navigate the different resources that we have. These are tags right here, and these are collections. And you will find here a collection called Getting Started. So I recommend for everyone to start right here. And the first course that I hope most of you have already completed is this course called Introduction to Bioinformatics. So Introduction to Bioinformatics is a course that is designed to give you this high-level overview of bioinformatics as a discipline and the way it is used in different domains. Domains like healthcare, biotechnology, and agriculture. Really, it's surprising to know that bioinformatics as a discipline has evolved tremendously and now is being used in a variety of different ways across all of these domains or industries. So this will give you a brief introduction with a little bit of history of how did we get here. So this is a place to get started, right? This is a place where anyone can maybe complete this course in maybe an hour or 2 or 3, right? So it's a really short course. The second one that I would start with is Bytes and Molecules. Now, Bytes and Molecules is a more expanded course, and here's why. It starts off by speaking about just this idea that bytes or data and molecules or biology are becoming more and more interconnected. And the reason for that is that we can study biology by using data. And as we study it more and more, we understand the potential that this approach to studying biology has as an impact on many different applications. The second course talks about citizen science, right? And what is citizen science? Citizen science is that everyone can participate in meaningful scientific discovery. It's not only a domain for professors who sit in, labs and do research. Because data is so widely accessible, because experts that sit in labs all day long at top universities like Stanford or MIT are generating this data and making it publicly accessible, all of us members of the public can access that data and make new discoveries, make new and interesting discoveries. So that's an important component here, that whether we are in research or just starting to get interested, everyone can be involved. And then from there, it starts building on these important concepts that you need to know to be able to analyze data. You need to understand the terminology. You need to understand where the data is generated, and then you need to understand what to do with it. And then we have a few introductory courses into genomics, transcriptomics, and metagenomics. And these are the types of omics data that you can leverage in your projects. So these two courses, by the way, you can see that all of these courses are free, right? So if anyone, right, you join this portal and anyone can start learning about all of these for absolutely free. So hopefully by the time that you are done with this program or this series of webinars, you have completed at least 2 free courses, Introduction to Bioinformatics and Bytes and Molecules. And this will already give you a lot. As you can see, we have introduction to different omics data and then applications, and you can even get started learning about programming languages and also get introduced to the T-Bioinfo platform, a platform where you can actually see how big data is logically analyzed and how to get some meaningful outputs. Okay, so how many of you have already started with Introduction to Bioinformatics and Bytes and Molecules? So if you have started or completed, you can write, something like completed introduction and started Bytes and Molecules.
The session for Bytes and Molecule holds tomorrow.
Okay, perfect.
So, but many have completed Introduction to Bioinformatics.
Okay, great.
We have about 70 or so participants who have shared their success with us.
Okay, excellent. Okay, so great. So if you're already completing or have completed Introduction to Bioinformatics, you know exactly what I'm talking about. And then excellent. Tomorrow we will have a webinar on Bytes and Molecules. Thank you, everyone. Excellent. Yeah. So if you have completed Introduction to Bioinformatics, again, what can you do with that? So you want to go to your profile, go under certificates, and find that certificate, Introduction to Bioinformatics, right here. You can view that certificate. Okay, this certificate is something that you want to share. You see, this is my certificate. I can share it. So how do I share it? I download it. I go right here and I share it with the community. Okay, sorry. The way to do this is to save it first as an image. I'm going to make an image real quick. And now I can share it. Okay, I can say, something like this, okay? So once I have this, I can also tag other people. For example, I think that a lot of people on our team have contributed to developing this course, and I'm going to tag some of those people right here, like Sonalika, Ojasvi, and Mohit. But because I'm here with you, I will also add a tag to Mr. Adekunle and Daniel. Maybe he's not here. Ayanfe. Okay, so now that I have shared this, people in this group are going to know about my progress, right? And so now you can see that I have completed this, and I can move on to next exciting challenges like the bytes and molecules, okay? And I'm sure that the recorded version will be shared with you as well. So getting started, right? After we get started, what do we do next? So getting started will be introduction to bioinformatics, bytes and molecules, getting started with bioinformatics in R, a programming language that is very useful in many aspects of bioinformatics, and getting started with bioinformatics in Python. So let me briefly then tell you about what else you are going to be able to do after you have completed these basic introductory modules. So the basic idea of this portal is to collect information that is kind of the foundation for becoming proficient and understanding the logic of bioinformatics in whatever project you end up going with. And we've organized it around the types of data. So you will see here genomics, transcriptomics, metagenomics, and skills. So for example, data science is a more code-oriented direction, or in some cases, we can look at it as a more logic-oriented, right? So not necessarily becoming an engineer and learning how to code everything on your own, but still understanding the logic of the analysis so that you can also use this analysis in an effective way. So really, we have two paths. One path is no code, where you understand the logic of the analysis. And the second path is code-oriented, so that you know how to start coding some of those logical steps and turning those steps into a project, right? And that's the third component, where we have essentially mentors and mentor-guided programs that allow you to develop an independent research project. So how can you learn bioinformatics without any coding? The way to do this is to get started with the T-Bioinfo platform, where you can process many different types of data that are found on the public domain and extract meaningful information so that you can start playing with that data and understand what it does. So for example, here we have one optional result from such a data analysis project, and this is a result for single cell sequencing. So something that is being used a lot today in research. Let's take a look at how this works, how this looks in real life. Now, all of you that have signed up are actually, you have access to the T-Bioinfo platform. So to get there, this is the link. Let me share that with you in chat. Okay, please take a moment to get to this link and try to log in. The way to log in, right here, you can see I'm logged in. But if I was to log out, I would click on login. And here I would use my email address to get in. Okay, so let me know once you're in, and we'll do a little practical thing together to see how this works. All right, let me know once you're here. Anyone? Okay, did anyone arrive at this link? All right, great. Thank you, Nono. Anyone else? All right, perfect. Okay, so once you are here, let's take a look at a simple pipeline, okay? It's going to be very simple at first, but again, in the coursework and in your future sessions, you will learn about more things like this. So yeah, there's no steps right now. We're just going to this link. So if you're here, great, yeah. So if you're on this page, again, you need to be logged in. If you have logged in, how do you know that you're logged in? You hover over this and you see your email address, right? So if you see your email address, that means you are logged in. So what we'll do is we'll go to this simple pipeline. We'll go to differential expression section right here, and we'll click on this down arrow button. And then we'll go into this Alzheimer's. Okay, so Alzheimer's disease is a condition where you lose cognitive ability, eventually actually leads to death, right? It happens later in life. But it's an important one because one question is, well, how do we know what is the cause of this disease? What actually makes this disease so dangerous, and how does it develop? So here we have an example of a simple project where we took a data set from the public domain related to Alzheimer's disease, and we want to compare people that do have the disease and people that don't have the disease. And if we want to compare those two groups, we can do what's called differential gene expression analysis. So we're looking at the difference of genes that are amplified in healthy and downregulated in Alzheimer's disease, or the other way around, amplified in Alzheimer's disease and downregulated in healthy condition. So to do that, we will use a job called DEC2. This is an algorithm that essentially looks at consistency across each group. And if the consistency is significant, so it is amplified across all samples within that group, and compared to the other group, all of the samples are downregulated, then it produces a p-value, which is an indication of this not being random, but something that is statistically significant, and gives us a value by how much, what's called a fold change. So how much more is it amplified in one condition versus the other? Now, when we find those genes, it is very useful to know that these genes exist, but it's not very useful if we want to understand what do they actually do. And to know what they do, we perform what's called enrichment analysis. Enrichment analysis basically looks at sets of genes, so groups of genes, and combines them according to their gene ontology or according to a pathway, a biological pathway that they are involved in. So we're going to perform this step. And we can also add on to that analysis by doing what's called gene set enrichment analysis. So two methods that kind of work together, and click on end. Okay, so what you should get is something that looks like what you see on the screen. And this helps you remember what these steps are. You can see that they are color coded. We start with a comparative analysis, DC2, and then we perform annotation. And this is enrichment analysis and gene set enrichment analysis. So what do we get as a result of this? We can see a few different outputs. So first of all, here you can have files. You can download these files onto your computer, or you can explore these visually. So you can explore them in a dashboard. You can view a volcano plot, or you can see here a heat map of top differentially expressed genes. What is a volcano plot? How many of you are already familiar with what a volcano plot shows? Okay. So anyone else? Does anyone, I'm sure, okay. Okay, so if you're not familiar with this, this is, okay, great. So some people are familiar with this already, right? What is a volcano plot for those who are new to this? A volcano plot basically represents, each dot here represents a gene, right? So the genes are dots, and they are positioned on this plot in this two-dimensional plane, right? You can see here the log2 fold change, and right here, the p-value. So you can basically think about this, the higher it is going up, the more statistically significant. And into each side, it is going to be more significantly expressed, so upregulated. And there's two groups, right? So there's a disease group and a healthy group. So if I compare disease to healthy, I will get some genes that are upregulated in disease, or some genes that are upregulated in healthy, right? So that's how you read this plot. So I have a group of genes, the red ones are statistically significant, upregulated for the disease, and these are statistically significant, upregulated for healthy. So that kind of helps me organize all of my data together. Now, this is an interactive plot. So if I hover over, I will actually see what this gene name is. You can see that this is gene called ATP binding cassette subfamily C member 12. And so this is a way to kind of bring in that annotation and understand what's going on with this volcano plot. Now let's take a look at another plot. This is what's called a heat map. So this heat map also shows us, if we look on the bottom, you can see that these are annotation of those genes, or of those samples, sorry. So the samples are Alzheimer's disease. So the disease samples are right here. And right here we have old. So these are just healthy old individuals. And you can see that some genes, right, are upregulated. So here, Rabfilin 3A is downregulated in Alzheimer's disease and upregulated in disease. Now, the dashboard organizes all of this information together and gives us some control over how it is presented. So we can actually change some of this visualization over here. For example, I can change the level of significance if I'm interested to kind of filter out some of those points, and I get right away the number of those genes at the bottom. Over here, I have the heat map, and I can select 20 top or maybe 50 top genes, right? Or maybe can limit it to 10, which will make it easier for me to see what's going on. I can also change the way this looks and maybe save this for my presentation or publication or something like this, right? Down below, I can also explore individual genes. So right here, let's look for a gene called VGF. Okay, and I can see that these two genes look the same. So they are downregulated in Alzheimer's, upregulated in healthy, right? So this allows me to navigate some of that information in an interactive way. I can also look at the enrichment plots, right? So the enrichment plots, as I mentioned, organize this information according to gene ontology terms. So biological processes are listed here, and the size correlates with how many genes are actually involved in this process. And right here, I also have KEGG pathways. So those pathways are listed right here. And again, the same way, I can now see that there are two main gene ontology processes and one main pathway. So the pathway is amyotrophic lateral sclerosis, right? Makes sense, related to brain function, cognitive function, etc. So if I want to now perform in-depth downstream analysis, I can also download all of this data by clicking on export, downloading the different types of data that I have right here, right? So again, what did we do? We understood how the logic of differential gene expression analysis helps us make biological interpretation of data if we understand the context, the project where this data came from, okay? How do we understand the context? Well, after you have completed the basic coursework, you can now explore different projects that we have on here. And the projects are data sets taken from the public domain that are curated and organized in such a way that you can perform these exploratory analyses on your own, understanding what's going on. And you can find projects on a variety of topics like cancer, neuroscience, infectious diseases, agriculture, and even space omics. So another part, right? So we talked about playing around with data, right? You have all of these different examples that you can kind of look at and play with. Another process is to learn how to code, right? So a lot of these methods that we just looked at, so we looked at, for example, how to create a heat map, or we looked at how to create a volcano plot, or we looked at how to perform enrichment analysis, right? All of this is done with commonly used tools in bioinformatics that are called packages. And these packages are typically used to produce figures, to produce standardized results, etc. So the way that you can start learning how to use these packages is by looking at some coding exercises that we have. For example, here, we have a course called Introduction to Data Science in R. And let's take a look at one common problem that people have, which is loading data and visualizing it, okay? Why is this such a problem? Well, most tutorials online, if you want to learn how to code in R, will start with, let's write hello world or something like that, right? But here we are performing some fairly complex analyses on, very specific types of data. And we would like to know how to do that, how to do something relevant to what we already understand and are interested in. So to do that, we are going to see an example. So in this example, we are loading some data, and we can run all of this right within the browser, right? So right here, I'm going to click on run, and it gives me an error. And that is because the objective of this practice is to fix the syntax of the code. Okay, so right here, I need to include table. And again, I have an error, and that's because you see the syntax is incomplete. And I have an answer, but it is incorrect. So if I follow all of the directions, right, and I actually look at what this says, I can start learning, okay, like this. Okay, the answer is still incorrect. So I need to read the directions here, but the idea is that I go from understanding how something is being used, right? So how something is leading to a biological insight. And once I understand that, once the concept is clear to me and I want to obtain the technical skills to do it, I can go back and learn how do people actually use this kind of coding to produce more and more complex visualization, analysis, interpretation, etc. For example, here we are learning how to make a scatter plot, box plot, histogram, and heat map, right? What we just saw being used right here in our ability to analyze these differentially expressed genes. So both sides are important, right? And what's important to do with this is to now organize the process, right? So all of you are now getting started, right? You're getting started with something that is fairly simple. And you are going to now continue to build on that foundation by exploring different types of projects, by learning how to practically run these kinds of pipelines and write code. And eventually, what do you do with this, right? So let's say you've got a couple of certificates. Let's say you've even learned something useful, right? And you've understood something. What to do next? Well, in my personal experience, I've realized that just having a certificate is not really that transformational. So for example, this is me. A few years ago, my son was still small, and I had a certificate from the Harvard Business School. And that certificate, over the years, I've obtained many different certificates for different types of coursework that I completed. And those certificates have proliferated so much. We now have Coursera, edX, Udemy, etc., right? So many different certificates. What is really the value? Well, what we discovered is that when you go to a potential collaborator, or if you're looking for a job leveraging the skills that you have, it is more important to have a portfolio, to say, I have created this project. I've analyzed this data. I have understood this subject and I know how to do it, right? To show that it's not enough just to have a certificate. It is much better to start building up your portfolio. So to help you build up that portfolio, we have these project specialization tracks in oncology, in infectious diseases, in neuroscience, and in agriculture. And in each one of those specialization tracks, again, there are things to understand, right? So things that you need to understand in the context, in the data, in the types of analyses, but then you become more independent and you can start asking your own questions. So to help you build out a portfolio, we have a special program called a research fellowship, or you can do this even on your own. And the way to do that is to reach 10,000 points, to complete a number of courses that we have on how to design a bioinformatics project. And after you have done that, you can actually submit a project proposal that our team will review and help you with, and then publish that project and make that project available under your profile. So let's take a look at some of those projects that others have done already. So again, these are practical courses. If we want to go and find a project, you simply click right here on project, and you will see many interesting project examples that you can start with. Or if you would like to start working on your own independent project, in your tab, in your profile, you go to the projects tab. You can see these are some project drafts that I'm working on. And right here, you can start a new project. right? So this is only going to be available once you reach a certain level. For example, you can see that I have 33,000 points, right? So it's over 10,000 points, and I have completed all of the requirements, and I can now start a new project. If you want to see other people's project, what have others worked on, you can click right here on projects and see all of the projects that our research fellows have been working on, right? So let's say I am a student and I would like to develop a portfolio of my own to share with a potential collaborator, to share with a potential employer, or to even know for myself that I have done something with the skills that I have obtained, right? I have learned about bioinformatics, but now I have done something with it. And it could actually be a very good first step towards a publication. So here we have many such people that have completed projects either independently or with a team, always guided by a mentor that can provide feedback on the progress of that project. And eventually, actually, some of these people went on to publish. So you can see a lot of these individuals. Again, you can click on this project, kind of understand what it's about. And you can also see the person's profile, right? And when you see the person's profile, you can connect with them on LinkedIn and see. So this project that she has completed is also now a publication. So you can see that she has a preprint and she also has a publication, right? So that is really, I think, the ultimate goal of this process. Not only have you learned about bioinformatics, you've understood the specific applications of bioinformatics in a particular domain, but you have also then shown that you can leverage what you have learned to design an independent project. So OmicsLogic is a community where we learn together, work together, and develop a portfolio. But what's next? What can you do with all of these achievements? What can you do with all of these accomplishments? So please write a comment if you can in the chat. What is your goal, right? Let's think a little bit about where do you want to go with all of this? You might be wanting to go into research, right? So for example, you're a graduate student, you would like to join a lab, you would like to pursue some specific project that you maybe already know what it is, or just exploring and looking for an interesting project that somebody else is working on. How do you go to a faculty member or a researcher or professor and tell them, I want to be involved in your project? And why should they take you, right? Well, looking from the faculty side, from the researcher side, typically, they are looking for people who can bring real value to the project right away, right? The whole concept of somebody coming and helping in a project is that they don't just learn about what you're doing, but they contribute and they make progress within the field that you're in. And so you're looking for people that have a demonstrated background of doing something useful. That's number one. And number two, that they already understand what you are doing, right? You don't want to spend hours and hours and hours to explain what it is that you're working on. You want someone that has already done the literature review, that already understands what are the unanswered questions, that maybe even has tried to attempt to answer those unanswered questions. So I would come to a researcher like that, and I would show them that I have done a project or maybe several in the field that I am interested in, that I have read the latest literature, that I have found the data sets that are available online, and I have explored those data sets. And now I become a top candidate for that person to hire me because I know and they know that we've done the work. Let's say I am looking for an industry career, right? If I'm looking for an industry career, what do I do with all of these skills that I have? Well, the first thing that I got to do is I have to figure out what kinds of jobs are out there for people like me. So here we have a listing of jobs on the portal. where you can start understanding what types of jobs are available and what are they looking for in terms of experience, skills, and background. So here you have academic jobs. Academic jobs are related to research. Maybe some of them are junior, like a research associate, or senior, like director of biotechnology. There are internships in bioinformatics. So let's say you have zero skills, right? You're just getting started, but you would like to understand what it looks like in terms of a real professional environment. So here you have jobs in the US, in India, and other places as well. Experience building. So experience building is when you already have maybe significant amount of skills, but you would like to build upon that and have more information, about where this direction could develop for you. So this is an experience building as well. So here you can search by country, you can search by category, and ultimately in each one of these jobs, you will be able to find what exactly are they looking for. So for example, sometimes they will say, you must have a PhD. Sometimes they will say, you only need a bachelor's degree and you only need to have like a year of experience. So let's look at how all of this ties together. So I'm going to skip the details. We have a lot of information on the different programs that are available, but let's look at how all of this fits together. So let me actually go to my first slide and let's summarize what we have learned today. And by the way, if you have any questions or comments, feel free to add those in the chat. So first of all, we talked about the role of data in research lifecycle, how there's continuing amounts of data being generated across many different domains, and there's really a gap. And that gap cannot be solved by a highly specialized group of people. It needs to be the job of everyone. So literacy in data analysis and interpretation is the domain of everyone who is participating in biotechnology, agriculture, and medicine. Number two, you can choose to become an expert, but even if you don't become an expert, you at least need to understand the logic of what's going on. And that's where it's important for everyone to understand how data is generated, conceptually, how it is analyzed, and how to interpret it. And sometimes that means you can develop a lot of data science skills, but at least you should know how to manage and analyze this data by looking at examples and understanding each step along the way. Then we talked about how to get started. And we talked about that it's oftentimes for people like us who already have a lot of experience, who have invested a lot of years or months into studying this particular domain, it is important for us to, not go back to the basics and write hello world on the computer. That's irrelevant. What is relevant is starting to deal with real data sets and making real analyses that are meaningful. And so OmicsLogic provides you with a clear and fast path to get there in a meaningful way. Then we talked about careers, how to get there and what kinds of careers are out there. So we talked about whether it's enough to just have a certificate and say, I know, I've read, I've listened to someone tell me about this, or is it more important for actually doing and demonstrating what you have done by developing a portfolio of projects? And then finally, we discussed the community, right? And so my question to you today is, what do you think we want to do to grow this community? So this community today has over 10,000 participants around the world. We have participants in India, in China, in Australia, in Africa, in the US, in South America, in Canada, in Israel, in Saudi Arabia, right? We have participants from all over the world. Now, just having those people is one step, but the next step is building out this community. So the community doesn't just, appear on its own. The community is somewhere where you benefit by taking something and you provide value by giving something. So first of all, how can you take? We have offered many resources that you can start using for free and eventually pay an affordable rate of just $10 a month to go all the way. We then provide affordable mentor-guided programs that you can leverage to build out your portfolio, experience, and develop a project. So that is how you can take. How can you give? Well, there are several different ways to give. First of all, you can give by becoming a mentor. So ultimately, after you have completed the training and have developed an independent project, you yourself can help others go through the same path. You can become a mentor to a group of students or work with our regional coordinators in Africa, in India, in the US to help others as well. You can also contribute project examples, right? So we have a few, but we would love to grow that. And you can get paid for that. So if you are an expert in the field, you understand some topic like agriculture or infectious diseases or oncology, we would like to talk to you about some project idea that you have that could be developed into a learning resource for others to use. And finally, you can also become an ambassador. If you think that this is an important domain and field, and you know of other students or researchers who need to learn bioinformatics and apply bioinformatics to their research, we would like you to be an ambassador for us at your university, in your city, in your country, whatever it might be. So our team is here, and if you would like to connect with us and discuss this further, we would love to talk to you about that as well. So I will share my email. If you would like to connect with me directly, I already shared my profile on OmicsLogic, and you've connected with me on LinkedIn. If you would like to contact me, email is probably one of the best ways and easiest way to do that. But also our team is here, and you probably know Adekunle, Daniel, and Fe, Sonalika, Ojasvi, and others who are here. So feel free to contact them as well. So with that, I will finish up my part of this presentation and turn it over back to whoever's going to finish up and maybe add on to what I just mentioned. Hello, Daniel? All right, thank you, everyone. Appreciate all the feedback and comments. And again, looking forward to connecting with you. I will definitely share with you the slides through our team. And thank you again for joining today.
Just pick up things to give something. Thank you very much.